prior to UMD, I had a small migratory operation. What I would do is I'd produce queens in the spring in North Carolina on the coast. Could usually get a end of February, beginning of March graft down there, and then do that until late April. And then I'd head up north to New York and do it all over again for a long summer. And I worked for Mike Palmer at French Hill Apiaries in Vermont during that same period of time. My business essentially got up to a point where I could either stop working for Mike and scale up or sell everything off and go back to school. And I chose to kind of do the latter. And one reason that I went back to school was there seemed to be a mix match between what I was observing in my colonies and Mike's colonies and what was being published about Varroa destructor. And really, I couldn't wrap my head around how was it that so few, relatively few mites were capable of such destruction in a large populous honeybee colony. And so what you're going to get here is essentially 2 chapters out of my thesis on viral transmission and mite behavior. So we're going to, and Varroa is the thing that we talk about more than bees. My hope is that we can essentially figure out the Varroa problem so we can go back to good classic fun beekeeping. But until we get there, we tend to talk about this pest a little bit more than we do about our own honeybees. So a little background, especially if you're super new. I'm assuming everyone here has some familiarity with Varroa destructor. If not, it's featured over there on the left. It's a small ectoparasite. It's a mite that jumped from Apis cerana onto Apis mellifera at least twice, and then spread globally. There's a couple unique things going on in this relationship. So deformed wing virus was in the honeybee population prior to the jump of Varroa destructor onto Apis mellifera. So this pathogen, for as far as we know, for all of bee history, was horizontally spread and vertically spread from host to host. So what do we mean by horizontal spread? That means within a generation, within the same generation, the pathogen can be transmitted amongst communicable modes. So oral transmission, where bees trophallaxis with each other or orally exchange food through brood food or something like that. We have these established horizontal modes. Now, once Varroa destructor leaps onto Apis mellifera, it turns out it can vector this pathogen. And we really want to highlight that this isn't exactly how all vector-host pathogen relationships work out. There are plenty of pathogens of hosts that can't be vectored by a vector. When we say vectored, that means the biting of some sort of, usually an insect, spreading a pathogen. So a good example would be something like tuberculosis. So tuberculosis, I can cough into your mouth, you can get tuberculosis, but a mosquito couldn't bite me and then bite you and then transmit tuberculosis. But that's exactly what happens with deformed wing virus and the mite and the honeybee. Mites feed on adult bees. They enter into these spaces in between the sternites, which you can see right down here in the center picture. Mites also feed on bees during their brood development. When they feed on a bee during their brood development, many of those bees will emerge asymptomatic. And by asymptomatic, we mean that we cannot see any overt infection on those bees. However, some of the bees will emerge with some sort of deformity. This could either be a shortened or truncated abdomen, or because of how the disease gets its named, these crippled wings. Now, these crippled wings happen when the mite's feeding on the developing larva into pupa. The virus migrates into the bee's brain. But not all bees are going to emerge this way. If you do start to see even a few bees emerging with crippled wings, especially this time of the year, it's indicative of having a really high mite load that's affecting your worker cohort. So Varroa go into those larval cells because they need to reproduce, and they feed on those pupa until they emerge, in which case they're released. If you haven't done this yet, it's actually really fun to pull out developing larvae with the mite on them. So mites will enter the cell before it's capped, and then your adult worker bees are going to cap that cell. That larvae very quickly consumes the rest of the brood food and then actually turns over in its cell, which is, so if you uncap that cell and pull out that larvae at the stage that you see in the center there, you can actually pull it out very often with the mite right on the larvae. So it's kind of a cool way to start to see some mites in your worker brood, is if you can find some recently capped cells. Now our bees will, and if you do that, use dull tweezers, just a cheap pair, not a sharp pair. It's actually really easy to pull them out if you use dull tweezers. Now our bees will also uncap developing pupae with mites inside of them. We've bred bees to engage in this behavior. So the act is uncapping and cannibalization. We called it Varroa sensitive hygiene for the longest time. And what we originally identified were some strains of bees were more likely to identify Varroa in their brood cells and release the mite so that it couldn't fully reproduce. And we've bred this as a way to attempt to control mite reproduction. So that was just really, really quick background. I'm going to jump into something else that's largely not talked about in Varroa and honeybees. So we've known for years that mites will move from one bee to another, but that behavior is largely not described at all. And in fact, if you were to look at the couple studies that have been done, that's essentially the description, that in petri dishes, they've seen mites move from an emerged bee host onto another adult bee. And there was evidence of mites moving inside of colonies, and that's it. And I thought this was really odd early in my PhD, because in other vector relationships, the biting rate is actually extremely important in disease transmission dynamics in those systems. And this has primarily been studied in mosquito relationships, and primarily in malaria. So in 1964, this guy named Garrett Jones comes out with a paper where he wrote this mathematical equation describing the vectorial capacity of mosquitoes. And the original equations, they're over on the left, and each of those symbols represents a main parameter that's important in disease transmission in a mosquito malaria human relationship. And by a parameter, we mean a thing that's important. So in this case, the proportion of the host is really important, and that's why it's in the equation. Now, the biting rate is the one that I highlighted right here. And the biting rate is actually overwhelmingly shapes the disease transmission of most mosquito pathogen relationships. It actually is overweighted in the equation. And so knowing this in mosquitoes, I wanted to know, does this apply to the Varroa and the honeybee and deformed wing virus? Because if it does, it seems like it might give us a better idea of how Varroa are killing our honeybee colonies, is if we could describe not necessarily how many mites are in your colonies, but how many bites are they taking in a given period of time. And this equation is all set on the number of secondary infections that would arise from one infectious host or one infectious parasite in a given day. So we knew we could modify it for the Varroa honeybee relationship. Now, since the 60s, a lot of work has been done on this, and one of my favorite papers was in Nature Communications, and it showed that the Pareto rule actually applies to malaria, and that there's always super spreaders in any mosquito population, and those super spreaders are responsible for driving most disease transmission. If you're not familiar with the word Pareto rule, you probably have heard of the 20-80 rule. So 20% of anything is responsible for 80% of something else. This usually comes down to some fundamental things, like 20% of your investments are going to be responsible for 80% of the value in your portfolio. something like that. 20% of your workouts is responsible for 80% of your strength gain, things like that. Well, it also applies in mosquitoes, where 20% of mosquitoes are actually responsible for 80% of infected hosts. So it's actually a minority of the vector population that's responsible for a majority of your parasitized hosts. Whereas there's 80% of these mosquitoes that are actually responsible for very, very, very few bitten people. And I was like, well, that's really cool. Does that apply to the Varroa and the honeybee? Sorry. And so if you are kind of conceptually thinking of it, if I have a group of bees and one mite, and because I am extremely skilled at graphic design, in this slide, you want to think that mites may be feeding on an adult bee, and they'll move from one bee to another in that group of bees. The rate at which they do that, we would expect there to be a huge heterogeneity or variation amongst the mites at the rate at which they move. And we found out with our research that there's actually a huge heterogeneity. So a little bit on methods, just give you a background because it's going to be important later. So whenever I did this work in the lab, we would do it in these plastic cups with hand-painted bees. I did groups of 8 bees for a couple of reasons. 1, it was a practical number of bees to run the experiment. 2, it gave me enough statistical power to do analysis. 3, 8 is not so many bees that it's not impossible to find 1 or 2 mites on those bees every single day with reliability. Whereas if we get up to 15 bees, it gets to be a nightmare, especially when the bees get older and they start to run around in the cages at faster rates. Whenever I was working with 2 mites, I would hand paint the mites different colors so that I could actually always track the mites separately from another mite. It's co-inhabited pest. And then anytime I used a virus in the lab, I would use this genetically modified virus that I could detect from any other strain of deformed wing virus. And that way, when I did my qPCR analysis, I could actually identify whether or not a virus that we introduced into our group of bees or into our mites persisted in our population. Whereas if I only used a wild type deformed wing virus, I would have no way of identifying my introduced deformed wing virus from background deformed wing virus that we're inherently going to have in our experiments. So this was actually a really handy tool that came from Eugene Ryabov and Jay Evans at the USDA. And again, our bees would just look like this, where we'd have 8 painted bees. And we just kind of track the mites moving from one to another. So now I'm going to jump right on to some results. So what I would do is I would run these experiments for 15 days, checking them every 12 hours. And so we get pretty good reliability of observing where a mite is, where it's feeding, and where it's not. So this graph over here on the left, as much as it's very, very boring, it's actually really important. So on the horizontal axis, I have time. On the y-axis, I have the number of parasitized bees. I still need to change that so that they're whole numbers. This is silly. Anyways, and we had some mites that died during the trial, and we had some mites that did not die during the trial. So mites that died are dark blue, mites that lived, or we call them survivors, who survived for the whole 15 days, they're blue, light blue. So the way to read this is if you're here in the bottom left, you parasitized very few hosts and you lived for a very short period of time. If you're all the way over here on the bottom right, you lived for a very, very long time. You actually parasitized no more hosts than the ones that died really early over here. Now, if you're up in this upper right quadrant, you're really special. Not only did you live for a long time as a mite, but you actually parasitized a lot of hosts. So mites up here parasitized 8 hosts, whereas ones down here who lived for the same period of time parasitized as little as 2 or 3. And so this was really important for us to see this data, because what it's essentially showing us is there's a huge heterogeneity in the feeding rates of Varroa destructor on adult bees. And this is the same thing that we see in the mosquito studies, where mosquitoes don't bite people at the same rates. Each individual mosquito either expresses low biting frequencies or high biting frequencies. And those high biting frequency mosquitoes are most likely to acquire a pathogen and also to transmit a pathogen. And you might ask, wait a minute, why would they acquire a pathogen? Well, not all of your vectors in any system are infectious. So in the case of Varroa, not all of our Varroa are actually harboring high levels or infectious levels of deformed wing virus. Some of these Varroa are what we would call naive. They may have virus in them, they likely do, but not at levels where they're going to be infectious when they bite a host. And this actually brings up an extremely important point in vector biology. Varroa destructor for deformed wing virus are called mechanical vectors. A mechanical vector is a parasite where the virus, or the pathogen, I should say, does not replicate inside of it. So the vector simply acquires it and passes it on, but there's no replication inside of the mite. So in vector biology, mechanical vectors, you can have a pathogen in you and you never transmit it unless the pathogen's actually at a really high level, usually in terms of viruses over log 10 values of 8. And so this is actually an important distinction that we should understand with Varroa, that they can be naive and they're not actually effectively vectoring a virus every time they bite one of your bees. But the ones that move at really high frequencies are likely to acquire a virus because they bite a super infectious host and they acquire the virus. Then they're most likely to move along and then pass it on to many other hosts. So that's why we would find these high frequency biters to be the most likely to drive viral transmission. This is actually the same data, just ranked, so it's far easier to read. And again, we get we're presenting it as the host switching rate. So the x-axis is actually just every single individual mite in the whole study, and the y-axis is the rate at which they switched. All the way up over here on the right, they're switching at 1 bee a day. These mites up over here on the right, and the ones all the way down on the left, they're actually switching extremely infrequently. And again, the colors, you can see that some of those live for a very long time, while some of them died before the trial ended. The data is not shown here, but there was actually no significant difference in time, the amount of time that a mite lived in any of the quartiles. So whether you were a lower quartile switcher or a higher quartile switcher, it didn't matter. Time was not actually significantly different between any of those groups. We did another study because one thing that we had to be really careful of was we didn't necessarily know that when a mite goes up in between these spaces that they're feeding every time they go up into those spaces. And what I ended up doing was I used these fluorescent microspheres. There are these 1 micrometer plastic particles that glow blue when we hit them with a fluorescent light underneath a scope. And I was able to get them inside adult bees. And then I put mites onto those adult bees. And then after I would see a mite go in between the space on the adult bee, I would capture the mite and smear it on the slide and see, did it acquire these beads? And they did. And in fact, mites are actually entering these spaces for the purpose of feeding. They're not resting spots. They're not just going on vacation there. They're actually going in there because they want to feed. So once we had this finding, like they're moving at different rates, they're actually going in there to feed, we really wanted to know, hey, what's the consequences of a mite bite alone versus a mite bite with virus, or no mite present whatsoever. And so I did this relatively large study. Again, these are all in plastic cups with bees that were procured from one single frame after they emerged. They're all hand painted. but I randomly divided the cups into 4 different groups. So if you were the really, really lucky group, you were an unchallenged mite, or unchallenged bee, you had no mite whatsoever. So you're scot-free, you're the mite-free cage. And if you're in an unlucky group, you had 1 mite added to your cup. Just 1 mite. So 8 bees, 1 mite, not that bad of a deal. But we would add 1. And some of those mites, we gave no additional virus. So we just collected a bunch of mites from a colony. We did this thing, what's called serial passaging them, to kind of clean them up, get virus out of them by transferring them from clean pupa to clean pupa. And the last pupa would either be a clean pupa or one that I injected with virus. So if you were a mite with no additional virus, you were just feeding on a pupa. If you're a mite in the VDV group, I actually injected the last pupa you fed on before the start of the experiment with Varroa destructor virus. You engorged on that virus, and then I moved you, I picked you up. And by the way, one of the easiest ways to pick up Varroa is just use your Chinese grafting tool if you graft queens. It's a great tool just to pick up Varroa with. It's a great way to handle them without killing them or damaging them. And I would put them into the cup. And same thing for deformed wing virus. I would use DWV for that. And so after I ran this trial for 15 days, checking mites every twice a day, every 12 hours to see where the mites were, collecting dead bees, collecting dead mites. This is the data we got. Now, I said if you're in that first group, you were really lucky. And I said that because you were really likely to survive. So if you were a bee in an unchallenged group, it was significantly different, your chance of survival versus any other bee in any other group. And there was no difference between the mite challenged or the mite challenged and virus groups, which are these 3 down here, all grouped together, of your chance of survival. So all of these were significantly different than our unchallenged groups. But whether the mite had virus or no virus, there was no significant difference between these 3 down here. You were just likely to die. Now we're going to break this apart a little bit as we go on, because in each of these groups, because I'm checking the cages every day, I know that some bees were never observed with a mite on them, and then I have some bees that were observed with mites on them. So we have 2 types of bees essentially at the end of the study that we can separate regardless of which group you're in down here. We have bees that were observed, fed upon, and we have bees that were never observed, fed upon. And in the challenged group with no additional virus, there was no significant difference in your chance of survival, whether you were fed upon or not fed upon. Which was kind of a weird result. It wasn't something that we were expecting right away. We kind of assumed, mite bite in and of itself is going to be bad. But that's the data we got. And we did get another pretty cool data, which was if you got bit by a mite in this group, you actually, for the most part, lived for a really long period of time. So there was a lot of variability in that. But some bees got bit by a mite and then lived for the whole study. They actually never died. And then some would die somewhat quickly, but for the most part, you lived about 160 hours after we observed you being parasitized by a mite. Now, if I was to go over to one of the viral groups, we get a really different result, and it's starkly different. So if a mite bites you with virus, you're very likely to die. You're not likely to live for a long period of time. And in fact, if you're in this group, there's a significant difference between your chance of surviving if you were not bit. And we saw the same thing in the deformed wing virus group. So whether the virus was VDV or DWV, it didn't matter. You were likely to die if you were observed parasitized by a mite, and you were really lucky there was a significant difference in survivorship if you were not parasitized. But this is starkly different than the control group, where there was no difference between the 2. And again, I really want to highlight and this is going to be important later, the bees that were bit by a mite in the control group lived for a long period of time, whereas all the bees that were bit in the viral groups died very quickly after being observed parasitized by a mite. I'm going to skip this because it's extremely boring. Actually, no, we can't. Okay, I'm going to really briefly go through it because I know a couple of you saw tables and I want you to have a good evening with me. And instantly there's like p-values and tables and everyone wants to run away. Look, I'm going to quickly go through it. Ignore the tables. We'll just talk about the stuff over on the right. Look, there's a thing called risk and relative risk, and it's really important in epidemiology. You most likely have seen a news article, likely in this month, where in some way they're referencing relative risk. And these are actually really popular because they're good clickbaits for us, and we tend to be really interested in them. All I did was apply something that we would do in human health and apply it to the honeybee. And risk is just essentially the probability of an outcome occurring. That's it. It just tends to be a bad outcome, right? Now, relative risk compares the risk of an outcome from one group that had an exposure to the very same exact group that didn't have an exposure. So this is where we're now comparing the relative risk between bees that were in the same group, but we separate them based upon fed upon and not fed upon. And I know what the data is over there. I'm just going to kind of skip it so I don't have everyone just kind of black out on me. But if you were bit by a mite with virus, you had a really high relative risk of dying. If you were in that group, you had a really low relative risk of dying if you were not bit by the mite. Good for you. But in the control group, you had a really low relative risk of dying if you were bit by a mite, which is good. But if the non-parasitized bees in that group compared to the other groups actually had a high relative risk of dying just because they were co-inhabited with mites and parasitized bees. And this is going to bring us up into our next chapter right after this, where we always have these other transmission routes of deformed wing virus in the colonies. And so why does the relative risk really matter in a honeybee colony? Well, ideally, I don't have mites and high levels of DWV in my colony, but we often find those two things together. Well, if a mite bit me and I was a bee, right, and I got bit and I died right away, that's really good for you because it means I have no opportunity to rear brood and pass a virus along. I have no opportunity to defecate in the hive and spread the virus through a fecal oral route. I really wish I wasn't using myself as an example right there with that specific transmission route. Or what's going to be predominantly important is with trophallaxis. Bees are constantly essentially kissing each other. They're using trophallaxis as a mode of communication, especially with the social gut in the colony. But they also use it for oral food exchange. And we know that transmission of DWV can occur in oral transmission. So if I die really quickly after I'm bit by a mite, I have very few opportunities to transmit the virus orally. But if I live for a long period of time after being parasitized by a mite, then I have ample opportunity to potentially transmit the virus by trophallaxis. And our results in this experiment suggest that this might be what's occurring. So these are log 10 values for DWV on individual bees in the trial that I just talked about. Now, unchallenged bees, those are the ones that had no mite. They were unlikely to develop high levels. They did not develop high levels of deformed wing virus in any of them. And the parasitized bees, the bees that were actually bit in the two viral groups were unlikely to develop high levels of virus, which is kind of the opposite of what we would expect. Again, remember, these are the ones that actually died extremely quickly after they were bit. The ones that were the parasitized bees that were likely to develop a high level of virus were actually ones that lived for really long periods of time, and they were in the control group. And non-parasitized bees in all of the groups, except for the unchallenged, developed high levels of virus. And I should say some of them did. Most of them didn't. Some of them did. And so we have a couple hypotheses out of this that we're working with. One is that the longer you live as a parasitized bee, the more likely you are to transmit the virus orally to your nestmates. And that's the one we immediately worked on. The second hypothesis that we will be working on is some of this data could just be because mites are avoiding bees that already have high levels of virus. And that's why those bees remained unparasitized. And to me, like that's a really cool notion to kind of investigate as a vector biologist. But anyways, that first hypothesis, that's what we really wanted to study. And that kind of leads us to chapter 2, which is, hey, I talked about this before, but deformed wing virus is both vectored by a mite, and it's also transmitted through these communicable modes horizontally from host to host. So, and this is actually really unique amongst pathogens. Let's see if the next slide is, yeah. So most pathogens on Earth are actually either vectored or transmitted communicably between hosts, but they're not both. So I'll go back to malaria and tuberculosis. Malaria is vectored by mosquitoes. If I get malaria, I cannot cough it into your mouth. It's not possible. It's not a transmission route for that pathogen. Whereas tuberculosis, we can readily transmit it person to person, but a mosquito can't bite me and then vector it to another person. It's not possible. Now, there are very few diseases on Earth that we know of that are actually both transmitted vector-borne and communicable. And there's only a handful of them. And so I'm going to highlight those here because I think it's really important for us to understand how unique deformed wing virus is and how much your bees are actually having to deal with when you don't manage your mites properly. So one of these pathogens is a virus called salmon isovirus. It's responsible for millions of dead salmon every year, largely because of the farm salmon industry, where we have these very concentrated cages in the ocean where salmon's farmed. It's vectored by this copepod, which you can see up here. It's also called a sea louse. But it can also be transmitted directly from salmon to salmon without the copepod. And that's from the mucus touching other salmon, or in these concentrated pens where there's really high density of salmon, the seawater can get so infectious that the seawater itself can be, I guess you would call it a fomite for other salmon to acquire the virus. The other one is Tembusu virus. Now, this is a virus that came from relative obscurity in the 1950s and is now spreading geographically. It's also called egg duck drop syndrome. It's predominantly found in farm ducks in Southeast Asia. It's vectored by a mosquito, but it can also be transmitted duck to duck. Largely through contact or fecal oral routes, or because of high density farming through like shared water troughs. And then the one that I don't have on here, the two, is West Nile virus that can also be transmitted host to host and by a vector. And now Japanese encephalitis virus has just recently been shown to have pig-to-pig transmission along with being vectored by a mosquito. And the one that we're all really, really familiar with is this one. It's not a virus, it's Yersinia pestis, the causative agent of Black Death, the plague. So Black Death is responsible for 3 pandemics in human history. The most well-known is in medieval times, where up to a half of the population in Europe passed away because of this pathogen. Now, fleas can jump from rodents onto people and vector the pathogen. If I get bit by a flea with Yersinia pestis, I get bubonic plague. But if you cough the plague into me, and I just kind of use cough the plague into me as a colloquial way to graphically describe a transmission route. not necessarily the words we would always use, then you would get pneumonic plague. But just like deformed wing virus and the other pathogens that I just mentioned, these all have multiple communicable routes and vectored routes, whereas again, most pathogens don't. And so if it sounds scary, I really want to insist it really is. We've had articles saying there's a plague in honeybees, and the media really likes this, but I'm actually doing a scoping review right now about essentially DWV being a plague of honeybees, because the disease transmission dynamics are very, very similar to the Black Death. And just like Black Death, DWV, or the other pathogens I just talked about, they're unstable relationships that not only kill on the individual level, but they also suppress the at the population level, where all of these pathogens have been known to cause up to 100% mortality in small populations. So in Tembusu and farm ducks, they've had entire farms wiped out. Well, we've had whole operations wiped out from mites and deformed wing virus as well. And there's some similarities in the hosts. They all tend to be really dense populations. So farm salmon, super dense, medieval London, super dense. Our apiaries are unnaturally dense. Even if you only have a handful of hives in your backyard, that's more colonies per square foot than you would traditionally have in the wild. And so I was really curious of, could we answer a couple questions about disease transmission in Varroa and the honeybee that haven't been answered yet or actually shown. So I do highlight this without purposely trying to pick on any researcher, but most of the disease transmission work that we have, that we know about in Varroa and the honeybee, come from laboratory experiments where the researcher either injected bees with a needle or fed bees with artificial food or tainted sucrose. But they didn't actually do it with having the bees or the mite do it. And so it was really important for me to actually run some trials where we were actually having mites vector a pathogen or having bees transmit a pathogen to other bees without intervention by us. And the main question that we really wanted to know was, could mites acquire deformed wing virus with the virus solely circulating amongst adult bees? Everyone's seen something similar to this before. The only difference here is I've added this little section right, oops, moved ahead on me. This little section right here, which is if this infected bee trophallaxis with this non-infected bee, and there's circulation of the virus through a group of bees, could a mite then acquire the virus from a bee that's never been touched by a mite before? Essentially, could trophallaxis act as a bridge maintaining DWV in the host population? And again, going back to that relative risk, those long-lived hosts are really important for maintenance of a pathogen in a host population. And by maintenance, we mean the continual transmission and the longevity of a host to carry on that pathogen. What would actually be really great is if a bee got DWV and just died, right? I know that sounds cruel, but then there's no risk for transmission to subsequent nest mates. So I set up this really cool experiment. Actually, I got to go back just to kind of show you. I forgot to put the slide in there. So I used to be a carpenter, and I taught myself in when I needed to in grad school how to cut up acrylic on a table saw and then glue them together, the pieces together with acetone. And I was able to come up with these what I think are really cool arenas where there's two compartments. They're ventilated. They each have their own sucrose feeders, but bees can trophallaxis with other groups of bees only through a feeder and not actually be in contact with each other. And so what we wanted to know is if a virus started over here, would it end up over here, and could a mite acquire the virus just through oral feeding? And sorry, we ended up seeing that. And I'm going to shorten this up just for time for everyone. But we introduced mites with a genetically modified virus inside of them and we had them feed on adult bees. And when we saw them feed on these adult bees, I would hand pick up the bees, which is pretty simple to do if you haven't done it. Just grab the bee by both of its wings. It can't sting you. And you can actually remove a mite from it with a grafting tool without harming the bee or the mite. And then I removed the mite from the study. But I kept the bee alive and put it back into a cage. And when that was all done, and I had a group of bees that were all parasitized, but now all the mites are removed, so they're no longer in the study, they're no longer around, I added recipient bees on the other side of the mesh screen, and we let them trophallaxis for about 10 days. And after that 10 days, I added naive mites, and naive meaning they have no genetically modified virus in them. We just grabbed them from the field and then introduced them into our arena, let them feed on these bees. And what happened was these mites actually acquired the genetically modified virus and then were able to vector it into subsequent hosts later on. And so we were able to show that actually trophallaxis can act as a bridge maintaining DWV in a population, and then mites can acquire the virus just from that maintenance between hosts. And so that was actually a really cool finding. And it took about 2 years to do that. There were a lot of hiccups that prevented the study from working. Some of it was just bad luck. Like one time my incubator spiked to 40 degrees, which essentially means everything's dead. Other times the virus just killed everyone so quickly that we couldn't do anything with it. And so when we finally got it to work, it was actually a really big deal. And then there was hundreds of hours of PCR work. Now, trophallaxis is not the only social behavior that your bees are engaging in. Early on, I mentioned that, hey, bees, we bred them to essentially uncap and cannibalize pupae to suppress mite reproduction. We've already published a paper showing that cannibalization of infected pupae can cause a super spreader event in adult honeybees. Well, I was really interested if mites could actually acquire infectious levels of DWV simply by feeding on adult bees which cannibalize pupae. So over here at A, I have adult bees that cannibalize infectious pupae. I take mites and then they feed on the adult bees. They were never in contact with these pupae. And then later we remove the mites and have them feed on pupae. And we actually find that these hosts over here, which used to be okay, then become infectious to the mites. So this mite that was not infectious actually acquires infectious levels of a virus and passes it on because adult bees cannibalize pupae. And so what we're highlighting here is we've largely looked at cannibalization of pupae as primarily and only as a good thing happening in our honeybee colonies. And it is good, you're suppressing mite reproduction. But our lab is now starting to highlight there are consequences and risks for bees engaging in this behavior. And I think probably the best way to put it is there's no free lunch. Your bees are going to cannibalize pupae and suppress mite reproduction, but the consequence is mites in your colony can actually become more infectious because of it. So again, there's no free lunch. And so we were able to show this whole route. The last thing, oh my gosh, is my slide not in here? Okay, it's out of place. So the last thing is we've talked about Varroa being dirty hypodermic needles. And I thought that was kind of interesting. And I was looking through the literature and there was very little proof of this. You know, we've called them efficient vectors for the longest time. And we said that mite sharing hosts can be sites of co-feeding and there's viral transmission between mites, but there was actually only suggestions of it, never proof of it. And so I said, well, I want to study this. And so what I did was I hand painted tons of mites, and some of them would be blue, and the blue ones got a genetically modified virus in them. And then some of them would be orange or green, and those were my controls. They had no virus in them. And what I would do is I would take a razor blade and open up the cell capping of a recently capped larvae. and shove 2 mites into the cell and then close the cell back up. And 8 days later, I would retrieve my mites. And then I would qPCR them to see if they acquired virus. And so in this cell right here, there's an orange mite and there's a blue mite. So those are our adult mites. They made a son and daughter in those cells. So that was kind of cool. And what we wanted to know is, did this blue mite, by feeding on that pupa, essentially pass a virus indirectly to the orange mite? And the answer is yes, they do. What we ended up observing were mites that entered a cell naive, not having virus, ended up leaving the cell having acquired a virus from the dirty mite that entered that cell. So we really could describe these as dirty hypodermic needles. And this is actually really important because at this time of the year, you're starting to see both more cannibalization in your honeybee colonies, and you're starting to see more co-infested hosts with more than one mite on them, both in the adult form and in the pupal form. So there's more opportunities for mites to acquire virus, and as we showed earlier in the multiple feeding, the relative risk to kill your bees quicker. And so that's actually the end of the viral work. Thank you for listening to the talk. And then I have about 10 minutes for Q&A.
Well, I have a question. That was super interesting. I'm new to beekeeping, and I have heard originally, like not originally, but earlier in the season, it's good to use the sticky boards under a screened bottom board, right? You know, to PAM it or whatever. So when the mites fall down, they stick.
Yep.
But then to take those off because it's warm and we want maximum airflow. So as the bees are cleaning themselves and the mites fall down, do they live long enough to climb back up into the hive? Like, what about that kind of mite life stuff? How long do they live not on a bee?
Yeah, so I don't know how long mites live off a bee. There's some studies and you get these averages. There's 2 things that are going to really occur when a mite falls from its host. One, it has the risk of desiccating. So if you're in a really humid environment, your mite's going to live for a longer period of time off of a host just because it's not going to desiccate. If you're in a very arid environment, and especially if it's windy and hot, that mite actually doesn't have very long. So some of it's going to be environmental. The second is mites actually feed at high frequencies throughout the day. So you can do a little study even in your own, kitchen, just have a warm box and take some mites and put them on adult bees in a ventilated cup, and then have some in a cup with no adult bees. And you're going to find within 4 hours, a lot of the bees, or a lot of the mites without hosts, are dead. It's not because they got cold, it's not because they desiccated, they actually starved, and they starved very quickly. So I don't have an answer for you, but I have those 2 tidbits, that it's going to depend upon your environment, and that they're also nutritionally, they get stressed really easily. I know in our studies, it's about 4 hours is the max that we can get out of mites with them surviving in an arena before they die. And then when we try to recover them by putting them on adult bees, very few of them recover. They actually die. And so, yeah, take that with what you will.
Cool. Interesting. Thank you. It's very fascinating.
Yeah, definitely.
A little maybe off topic from your research, but you were talking about the different ways deformed wing virus can vector. How long can it persist? So if a sick bee puts nectar in a cell, how long does that virus survive? Or can it be viable? Like another bee can go lick that up a few minutes later, or hours, or?
Jeff, I think this is like an unbelievably great question, because it gets into something that a lot of people ask, and also a fundamental thing in like human health and all disease transmission. there's almost never 100% efficiency in disease transmission. If there was, we would all either need rockstar Iron Man type immune systems, or we'd all be dead. It'd be one of the two things. And so with deformed wing virus, we don't get 100% efficiency transmission every single circulation. So let's use the example that you just had. If there is a bee harboring high levels of deformed wing virus and it deposits some nectar in a cell, it would have to essentially transmit enough virus while storing that nectar that virus is in a high enough level to infect another bee that ingests it. And so there's the first speed bump into transmission efficiency. So if it's transmitting a little bit of virus, that nectar is likely not infectious. And if it's doing a high level of virus, it could be infectious. Now let's think about the things that make something infectious. Well, the other half of that's the host, right? So if I'm a host and I have a really bad immune system, or I happen to have a parental line that didn't give me the genes, for great viral immunity, then I might be what's called susceptible to the virus. Whereas you might have the better paternal line and you can ingest a lot of that virus and you don't get sick, right? So we have susceptibility to the host that could either foster transmission or impede transmission. And then we also have host behavior, right? Hosts, we've seen from other people's work, can avoid pathogens. So we don't know actually if those hosts might choose to avoid that infectious nectar for a while, or just ignorantly slurp it right up. The next thing is, we often call it just luck in our lives, but that no one touches that nectar for one reason or another while it's actually infectious, if it even is infectious. And this comes down to something like the amount of time that something can remain infectious. And we don't know that answer. So there's going to be like a lot of variables at play. And you let me riff on them a little bit, but I want to come right back to that transmission efficiency. So in our work, when we started with infectious mites in any trial, we would see a really high transmission efficiency, but it would always go down after every single circulation. So if we started with an infectious mite and it fed on a bee, and then it went to another bee, and then it finally went to mites and a pupa, it would keep going down.
Yeah.
And that was really cool. But our experiment was not set up to study transmission efficiencies. To do that, we would need to have like 10 times the size of the trial. And that's thousands, tens of thousands of dollars more than I had for funding. I was really well funded by Pam Costco, but I wasn't that well funded. But to me, that would be like a super fun thing to study, because then we can really turn around to you and say, hey, here are the things that are really infectious in your colony. And not only are they infectious, they're likely to cause a lot of subsequent infections. I would point out that things like nectar are likely not to be super infectious. Even though we've seen that DWV can be in pollen and it can be infectious, we don't know the efficiency of pollen to adult bee infection rates, and it's likely far, far, far lower than just having infectious mites feed on you.
Great, thank you.
Yeah. Cool. Well, if there's another question, feel free. If it's on management, I'm happy to answer that too. You're catching me at the end of my night because I'm on East Coast time. So sorry for the relatively dry talk, but
I think you blew our minds. Lots to think about. Thanks for presenting.
Okay, well, thanks for having me for your group, and I hope everyone has a great fall. And then, oh, I should mention, because it wasn't in this particular talk. Actually, I can just quickly show this because it does matter. Let me just scroll right here. Hey, this actually matters for this time of the year. Actually, I'll just take 2 seconds because it actually goes to the multiple feeding thing. Hey, I did this field trial where we wanted to see mites moving from one bee to another, but it's impossible to track one mite in a colony. We tried, it's just not possible. But we can actually track bees far, far easier just by hand painting them. Sorry. And we set up this really relatively easy, but very, very tedious experiment where as worker bees emerged, we would pick them up and look for mites on them. And if they had a mite, we would paint them a couple colors. And if they had no mite, I just gave them one color to denote their age. And at the end of the experiment, I essentially get 3 groups of bees. I get never parasitized bees, bees parasitized during their larval development, and we have bees that were parasitized during adulthood. And so the way I like to describe this is if a mite bit you as a bee and you glowed in the dark, then we'd actually be able to see how many bees in a colony were bit, not necessarily how many mites there are in a colony. And there's 2 really different things. And so if you had a mite-free colony, this is what your colony would look like. It's tons of non-parasitized bees trophallaxing with each other, depositing nectar like Jeff mentioned, rearing brood, just all non-parasitized. But in the late season in our trials, so in August, so this time of the year, right out like what you all are dealing with right now, this is what your colony actually starts to look like if it has any high mite level at all. So the brown bees were never parasitized. The green ones are parasitized during development, and the pink ones are parasitized during adulthood. Now, our alcohol wash was close to 15%, so this was a heavily infested colony. But the effective or absolute parasite burden was close to 60%. What that means was, so relative parasite burden, that's essentially your alcohol wash. That's how many mites are over a portion of hosts. And so we often just say X over 300, right? But my effective parasite burden is, given the same number of hosts, how many of them were actually burdened by a parasite? So how many of them were observed bit by a mite? And it's actually the majority of the colony. And this work was done in 9 days. So within 9 days, 60% of the colony was bit by a mite. And that's what's happening in your colonies this time of the year right now. And so if I could leave giving any, I'll stop share for a second. If I can leave giving any new beekeeping management advice, if you have colonies that are at threshold or above, you need to treat now or manage your mites now. Don't wait for the quote unquote fall, because we're observing in colonies in 9 days, the majority of your bees can be bit. And as far as we know, there's nothing good about being bit by a parasite, right? Like no one wakes up in the morning and goes, I got a tick on me. Did you see this? Like no one says that. No bee gets bit by Varroa, who are becoming more infectious at this time of the year, literally because of cannibalization and co-feeding, getting virus bitten them and said, I think I'm going to live through the whole winter. So, I mean, and we see time after time as we do these experiments that these colonies just crash on us. And so if you have a super fall flow, okay, great, but do your mite washes now. And if you're getting really any detections, you've got to give up that flow and treat. And if you were going to treat, but you really wanted to push it off to a broodless state, I got a warning for you. Your whole colony's bit before you actually get to that treatment. And so even though best management practices haven't caught up to like the new findings that we're having, we're already changing our management over here because we know it's going to impact our survivability for the winter. And now it's in line with the vector biology of this parasite, which is, hey, we used to give suggestions based on, relative parasite burden, but we have this new data showing that much earlier in the season, all your bees are getting bit essentially, you should probably treat before that actually occurs. So
that last slide was really interesting because you didn't touch on the drones, but it was what 97% drone infection.
yeah,
so if we're
on that,
breeding queens, you have your late season queens that are going out. They're getting inseminated by sick drones.
yeah, so that's outside of my scope of studies. I did try to do some of that work. So there's one error with that reporting right there on that last slide with the 97% of the drones infested. At this time of the year, your colonies have very, very few drones. So that colony had 59 drones in the study, which was far, far less than I think the 400 something workers we did detections on. So when you have a small sample size, it's really easy to get an artifact of a really high infestation because you have it's like if I pick up one fish and I find that one fish is a trout, I can't turn around and say 100% of the fish in the lake are trout. And that's essentially what we got there. But Jeff, you bring up this really, really, really important thing. And it's unfortunately what our work is showing is that so many drones can get parasitized by mites that it looks like the majority of the drones going out for your mating flights have actually been bit by Varroa. Now that enters into queen reproduction biology and mating biology, and I don't do that. I do viral transmission. And so, and unfortunately, vertical transmission is actually I think it's far more complicated than horizontal transmission. And I'd be kind of stepping out of my lane to do that work. And so I shared it with someone, this guy, Garrett at Purdue, because I think it's something he's going to end up working on, but yeah.
Yeah, because I mean, when we think of people sharing bubonic plague with each other, I mean, that makes a lot of sense. But if your mother is sick, that adds a whole new factor into everything.
Yeah, it really does. So I mean, and we've seen that, study after study, drones have this high variability in sperm viability. We seen from Almiri and other people in Europe that drones can have really high infectious levels of DWV in their semen. The cause of that, don't know. I was talking to someone who said it's probably unlikely because of the developmental stages that being bit during adulthood is causing them to be infectious, but during development likely. I'm like, oh, okay. But again, that's outside my lane, and I'm pretty fixated on the couple things I'm working with. But I think the distinction to bring up, Jeff, is when we talk about Varroa feeding on workers, that's affecting the colony that they're within. Whereas we talk about parasitism on drones, that's going to affect the colony they mate with, or essentially the queen for the colony. That's another one. But yeah, are you a queen breeder in your area?
I raise queens, yes. Yeah.
Oh, you do? Okay.
Not a big, not commercial in any way, just for myself and some other.
No, but you're dealing with this.
Yeah.
So yeah, you're absolutely dealing with this. You could produce the best queens you want. You're still reliant upon your neighborhood.
Mm-hmm.
So cool. Well, everyone, thank you very much. And then, yeah, keep in touch. I think I'm going to be out in your area. I got to talk in Colorado, and I think I'm just going to drive out there because then I can climb for a couple weeks. And then my brother lives in Portland, so I'm just going to probably zip out and then climb a bunch of the Cascades and then drive back.
Cool. It's a good time of year to go up.
Yeah.
Well, if no one has anything else, I think we can wrap up, and thank you for the great presentation. It's so quiet when it's muted.
Well,
thank you.
Thank you. Appreciate it, everyone. Have a good one.
Yep, thank you very much. That was interesting.
Thank you.
See ya.